All right, whenever you're ready.
My name is Eleanore, and I will be talking about animal cloning. On July 5th, 1996, Dolly, a clone of Maple Dorset sheep ewe, was the only successful birth born out of 277 embryos that were made by somatic nuclear transfer. She lived until she was 6 years old when she died of progressive lung disease. Somatic nuclear transfer is conducted by taking the nucleus of a mammary gland tissue cell of one female and inserting into an egg of a different female that had its own nucleus removed. The fusing of the two occurs when an electric current passes between the two cells. This fused egg is then placed in a third female for the remaining gestation period. The sheep nuclear transfer experiment was first introduced by Steve Wilderson. Dolly was from an experiment conducted by Dr. Ian Lemieux and Dr. Keith Campbell at the Roslin Institute in Edinburgh, Scotland. They believed that this cloning would have a greater successful rate if the cells being donated in the GO, a specialized stage of cell division, where the cell is both susceptible to reprogramming and the nucleus is still a diploid cell. Animal cloning is a sub-area of genetic engineering. Since Dolly, scientists have been able to clone horses, cows, cats, rats, and dogs, but have had a low success rate. My claim is that animal cloning will fail to produce significant benefits. I will prove this by 1, animal cloning is not cost effective, 2, animal cloning will negate genetic diversity, and 3, the byproducts of cloned animals will have a low impact on human medicine. My first point is animal cloning is not cost effective. A cloned animal can cost, depending on the species of animal, from $15,000 to over $200,000, which limits the amount of people that can access it, and thus the need for it is low. It is also not cost effective because of its high failure rate of less than 5% of clones successfully being born. These animals that are successfully born have shorter lifespans than their parent clone, and some have developmental problems such as kidney disease. Aaron Levine, an assistant professor in the School of Public Policy at Georgia Tech, says in his book Cloning, among embryos that implanted start to develop, the most consistently observed defects relate to placental development. He also says that although scientists are optimistic, conducting the large-scale studies required to understand gestational losses during cloning is an expensive and long-term project. So with the high cost to low result ratio and the need for long, expensive research, animal cloning is too out of reach to produce any benefits. My second point is that animal cloning negates genetic diversity. With the changing world around us, cloning animals will slow or halt animal genetic change that happens through two parents passing half of their genetics to their offspring. Emma Hawkridge, who has a master's in sustainable development advocacy and is head of policy for the Soil Association, is quoted in an online article titled Cloned Animals, A Threat to Genetic Diversity, saying that given the current expense of cloning, it is likely to be limited to high-yielding dairy cows to maximize yields, and as such, in the short term, it's only likely to reduce genetic diversity. Genetic diversity helps to prevent against diseases and also keeps animals from inbreeding and from having one superior animal sire many offspring. Also, due to some clones being bred normally, they can pass on their shorter lifespans. This shorter lifespan is due to shorter telomeres, which are nucleotide sequences that protect the end of the chromosome from deterioration or fusion with neighboring chromosomes. Because telomeres deteriorate with increasing age, clones usually live half of their parent clone's lifespan. So with limiting genetic diversity by using a small amount of parents for clones, animal cloning will not produce benefits. For my last point, the byproducts of cloned animals will have a low impact on human medicine. Scientists say that the organs produced by clones can be used for transplant. What they fail to say is that clones tend to have a large offspring syndrome. The Learned Genetics Center at the University of Utah says that clones with LOS have abnormally large organs. This can lead to breathing, blood flow, and other problems. There is no way of telling whether a clone will have LOS or not, and with such a low rate of successful clones, animal cloning impact on human medicine will not have many benefits. In conclusion, I show that because animal cloning is so expensive, the need to clone animals will be low, and thus the research unneeded, that having the same clone siring many offspring can negate genetic diversity and encourage inbreeding, and cloned animals will not provide many organ transplants and thus not have a large impact on human medicine is why animal cloning will fail to have significant benefits.
All right, well, I like the use of Dolly as an attention device and introduction of the subject. You do a very good job identifying a specific claim and identifying that it is the proposition of the speech. There's an excellent layout of what the supporting structure is. In the body of the speech, you also signpost the supporting points very clearly, and you make pretty straightforward transitions on those points. Sometimes the delivery is a little bit problematic because you are going very quickly, and when you go through those transitions without taking a pause, without looking up at the audience, the material could run together a little bit. People who are listening closely, they'll understand and they'll figure it out. But remember, we have a lot of casual listeners in the world, and listening is one of those things that requires a lot of effort. You can't count on your listeners always making the effort, so you have to sometimes help them out a little bit more. I thought that you had some good quotes about the limitations on cloned animals, particularly on development and on diversity issues. There was a little bit of factual data that you presented, the 5% success rate, the half-life for the animals later on, that I didn't always hear source citations on. The cost issue, the same sort of thing, how much it costs to clone a single animal. I know it came from someplace, but I didn't get a citation on that. But the inferences made by the experts as to what that means in the long run for either research or marketing, I thought that those quotes were usually attributed very clearly. I thought you had a very effective summary at the end of the speech. There's too much reading, no audience contact. It sounds like you're rushing, so the delivery is a little bit of a problem. But the structural issues, most of the evidence, and the clarity of the claim is helped quite a bit, and it allows us to follow the argument, again, if we're listening carefully. All right, thank you.